Genes are composed of bases of DNA in long strings, and those strings need to be replicated.
DNA has 4 bases that are called A, C, T, and G. And so these bases have a ribose, which is a sugar, that's connected by a phosphate to an additional base. They then have the base, the individual A, C, G, or T, that's connected off. And so A, C, G, and T have specific patterns of hydrogen bonding that enable A to bond to T and C to bond to G. And this gives you the classic double helical structure of DNA that you've all seen.
A genome is the set of instructions contained in your DNA in every cell in your body. And that set of instructions contains the information needed to basically build an organism. But one of the really amazing things about genomes is that this entire set of instructions is contained within every single cell of your body. One of the most important components of the genome are the protein coding genes. So they're the genes. They're the ones that encode instructions on how to make up the proteins of which life is composed, of which cells in your body are composed, and the cells make up different organs, and that all these different components make up the organism. The genes are sprinkled throughout the genome in various places.
These days, once a genome is sequenced, we then have the whole set of genes. And so, for example, many mammalian genomes have 20 to 25,000 genes. Transcription is the process of making an mRNA molecule from a piece of DNA. The mRNA molecule itself is then spliced and, through the process of translation, turned into an ultimate protein product. And so the concentration of the protein product, as well as what the protein does, reflect the function of that gene. The process of transcription is the process by which transcription factors, both those that are specific to an individual gene, as well as a general transcriptional apparatus, binds to DNA and causes the DNA to be copied over into mRNA. mRNA is a molecule that looks very similar to DNA, it just has an extra hydroxyl group. And so this mRNA copy of the DNA can then float away from the chromosome and bind to a ribosome, where it then becomes translated into a protein. And so there are additional RNA molecules called tRNA molecules that are involved in this translation process on the ribosome. So the genetic code by which we convert a DNA sequence to a protein sequence is a triplet code. And so we have 4 different DNA bases that form 64 different codons. And so these encode 20 amino acids. And so there's redundancy to the genetic code. And so each codon, or each piece of 3 nucleotides from the DNA in the direction of DNA to protein, uniquely identifies an amino acid. Both metabolic pathways and signaling pathways require collections of proteins to function together.
We're at a real revolution in our power to sequence large amounts of genome data, right, with the next generation sequencing technologies that have come online recently. CRISPR technology is really changing almost everything about how we study biology. It has opened up whole new fields. It's one of those revolutionizing technologies that, in really providing us with the ability to edit genomes at our fingertips in a very efficient manner, in my opinion, we're really at the cusp of a revolution that in 10 years we're going to look back on these early days and think, oh my gosh, why didn't we see the implications of this? So it's got such far-reaching, wide-ranging implications that I think it's almost impossible to imagine all of the ways in which it's going to revolutionize, molecular technologies. Because we have these amazing advances in technology, and basically we're at the cusp of a genomic revolution, both in the amounts of sequence data that is being collected now, as well as our abilities to go in and edit it, to recreate it in the laboratory. It's a very exciting time, in my opinion.